So we're going to take a look at Chapter 2, the chemical context of life. And so the chemical connection to biology. Biology is the study of life, and organisms and their environments are subject to those basic laws of physics and chemistry, those physical sciences. One example is the use of formic acid by ants to protect themselves against predators and microbial parasites, as you'll see here in this following picture. Okay, matter consists of chemical elements in pure form and in combinations called compounds. Organisms are composed of matter, and matter is anything that takes up space and has mass. Matter is made up of elements, and an element is defined as a substance that cannot be broken down to other substances by chemical reactions. A compound is a substance consisting of two or more elements in a fixed ratio. A compound has characteristics different from those of its elements. Here you have a picture that shows sodium, which is a soft, silvery, shiny metal that you can actually cut with a butter knife. It's reactive in oxygen, and it'll actually catch on fire here fairly shortly if he leaves it exposed to oxygen, and eventually it'll probably explode if he doesn't handle it properly very shortly. But generally, we keep it stored in oils and such to prevent it from having exposure to oxygen. It likes to react with oxygen violently. So on the right side here, you got chlorine gas. It's a yellowish green gas as it exists in its natural state, and again, chlorine will react with water in our lungs and form hydrochloric acid, which of course will kill you, and it's been used as a chemical weapon before, I think back in World War I. However, these two dangerous elements, sodium and chlorine in their natural states, when put together and reacted, will form something entirely new, sodium chloride, table salt, which is perfectly fine for us to eat, and we do. We use it all the time on our foods, pizzas, french fries, etc. But any of these elements, independent in their natural state, would certainly do us in short term. So some pretty neat examples. Okay, the elements of life. About 20 to 25% of the 92 natural elements are required for life. These are called essential elements. Carbon, hydrogen, oxygen, and nitrogen make up 96% of living matter. Most of the remaining 4% consists of calcium, phosphorus, potassium, and sulfur. Trace elements are required by an organism in only minute quantities. That's why they're called trace elements. Okay, here in this table, you can see the elements in the human body. On the left column, you have the elements, and the middle column, the symbols, and the percent of body mass, including their constituency in water. So you have oxygen, carbon, hydrogen, nitrogen making up 96.3%, and then calcium, phosphorus, potassium, sulfur, sodium, chlorine, and magnesium making up another 3.7%. You can see you add that all up, you get to just about 100%. And so these are the supplements you generally see in like dietary supplements that we consume to give ourselves a little extra boost, or if we're not getting enough of a particular element in our diet, we can certainly get it through a supplement. And these are things that we need for proper cell and tissue functioning. So they're important to have, and you can see the four biggies here: oxygen, carbon, hydrogen, and nitrogen make up 96.3%. So those four are very important to us. Okay, some elements can be toxic. Some species can become adapted to environments containing these toxic elements. For example, some plant communities are adapted to this plant called serpentine. And here it is growing on this coastline, and I'll let you read about that in the textbook. You can find out about some of its unique characteristics in there, and I'll leave that exciting dialogue or reading up to you. An element's properties depend on the structure of its atoms. Each element consists of unique atoms. An atom is the smallest unit of matter that still retains the properties of an element. Can you break an atom down into smaller parts? Yes, but at that point, you no longer have that property of an element any longer. You start having individual properties of those subatomic particles, as they're called. And these subatomic particles are neutrons, protons, and electrons. And yes, these can be further broken down as well. We're not going to get into that for this course, but know that's in fact the case. But primarily, these parts that make up the atom, as we're concerned as biologists, are neutrons, protons, electrons. Neutrons have a neutral charge, as indicated by the name, no electrical charge, neutral, neutron. Protons have a positive charge, and electrons have a negative charge. Neutrons and protons form the atomic nucleus. Electrons form a cloud of negative charge around the nucleus. Neutron mass and proton mass are almost identical and are measured in daltons. Now, the electron is so small in comparison to the neutron and the proton that we generally don't calculate its mass. because it's at least 4 orders of magnitude smaller. So here on the left side, we have a cloud of negative charge, right? That's that yellowish area. And then inside of that, they're depicting this disproportionately large nucleus with 2 protons in the pink and 2 neutrons in the brown. But in reality, you can kind of see the electrons. They're showing them there in an orbital. There's 2 electrons with 2 negative charges to balance out the 2 positive charges of the protons in the nucleus. And it looks like they kind of go on this track around the nucleus. That's not, in fact, the case, but for intents and purposes, as far as diagramming this stuff, it kind of makes it easier to keep these things in these simple little orbitals. Okay, atoms of the various elements differ in number of subatomic particles. An element's atomic number is the number of protons it has in its nucleus. If you change the number of protons, you change the atom. It's no longer going to be that element any longer. Okay, that's the only thing you can't change. You can adjust the number of neutrons. We call those isotopes. You can change the number of electrons. We call those ions. But you can't change the number of protons in any feasible fashion as far as we're concerned in biology. The chemists and physicists can do some stuff in particle accelerators perhaps and stuff, but we're not going to concern ourselves with those types of issues for what we do in biology. An element's mass number is the protons plus the neutrons in the nucleus. So if you have 2 protons and 2 neutrons, that would have a mass number of 4. Atomic mass is the atom's total mass. That can be approximated by the mass number. All atoms, so when we talk about isotopes, for instance, all atoms of an element have the same number of protons, but can differ in the number of neutrons. So if you look at helium, for instance, all helium atoms are going to have 2 protons. That's what defines it. That's what makes it be called a helium atom. It has 2 protons. Now, if you change the number of neutrons, you can do that. That is still going to be helium as long as it has 2 protons, but we call that now an isotope. Isotopes are 2 atoms of an element that differ in the number of neutrons they have. Radioactive isotopes decay spontaneously over time. They give off particles, they emit particles and energy over time, and they eventually will, over some period of time, depending on, it could be seconds or minutes or hours or days or millions of years even, decay into a different element over some period of time. But we'll talk about that a little bit later. Radioactive tracers. Radioactive isotopes are used as diagnostic tools in medicine. They can be used to track atoms through the metabolism, so you can use a radioactive tracer like in a hospital diagnostic type setting, where you could give an individual a radioactive tracer and maybe a drink or an injection, and it'll go to a certain part of the body, and because it's radioactive, we'd be able to see it through a diagnostic piece of equipment, MRI or X-ray or whatever, just depending on what it is and how you're doing it. But there's a variety of different tools, and you can see how it's acting in the body and where it's going and what it's reacting with. So they're very useful tools. You're probably familiar with them or know people have had them or used them or done it yourself. But those are called radioactive tracers, and we generally want to use stuff that's got a short half-life in a hospital. You don't want to obviously use something that's got a half-life of millions of years, because it will the patient will obviously long be dead. We all will be long before you can get your results. So you want to use something that's got a very short half-life in a diagnostic setting, something with minutes or hours, or at the most maybe a day or two. They can also be used in combination with sophisticated imaging results, as I mentioned there, X-rays or MRIs or who knows what kinds of equipment they have in hospitals now. I don't know all the names, but certainly equipment that can detect these radioactive tracers in the body. Okay, and here's a picture of how one would work on an imaging tool in a hospital. So they've utilized a radioactive tracer to mark out a cancerous throat tissue here, and here's the image in which it's showing up. And so that's kind of what you would see as the radioactive tracers all are attached to something on the cancer that will cause them to stick to that specific part of the body and emit an image. Okay, radiometric dating. A parent isotope decays into its daughter isotope at a fixed rate. This is called the half-life. I mentioned this a moment ago. It's the amount of time it takes something to decay into half of its original parent isotope. In radiometric dating, scientists measure the ratio of different isotopes and calculate how many half-lives have passed since a fossil or rock was formed. Half-life varies from seconds or days to billions of years. Energy levels of electrons. Energy is the capacity to cause change. Potential energy is the energy that matter has because of where it's located or structure. So picture a book sitting on like the edge of a table where it could potentially fall off. That would have a lot of potential energy. It's not falling yet, it hasn't been converted to kinetic energy, but it has the potential, so it's potential energy. The electrons of an atom differ in their amounts of potential energy. An electron state of potential energy is called its energy level or electron shell. So they use this diagram here. On the right side, you have a ball bouncing down a flight of stairs. It can come to rest only on each step, not between the steps. What they're trying to depict here is that these energy levels exist only in specific regions. So for instance, if you have your atomic nucleus here in the lower diagram and the big gray ball there at the bottom of the screen, and you have these circles, these rings that go around it, each of these are depicting an energy shell, and the further you go out, the more energy is required to be in that orbital. So as you go further and further out, energy is being absorbed. As you jump back in, you're releasing some of that high level of energy to drop to a lower energy level as you move closer to the atomic nucleus. Now, what they're trying to make point of here with the ball dropping down the stairs is that you cannot be anywhere between these rings. You can be on one of these rings, but you can't be between it. So for instance, if you have ring 1 and ring 2, the innermost ring and then the middle ring there, call that number 2, you can't be at like 1.5 or 1.8 or 1.3. You can be on 1 or 2. Nowhere in between. So you either have enough energy to get to the next energy level or you do not. Even though you may have some accumulated amount somewhere in between, that's not going to bump you to that next energy level. The chemical behavior of an atom is determined by the distribution of electrons in electron shells, and the periodic table of the elements shows the electron distribution for each element. So here's a periodic table. Now they're showing you how it's depicted here. You have the first shell, which has hydrogen and helium, and hydrogen has 1 electron in its innermost shell, and then helium has 2 electrons in that innermost shell. And then when you drop to the second row of the periodic table, you add another shell on, another row of another orbital, if you will. So then you start filling that one. So the first ring only can contain up to 2 electrons, and then the second ring can contain up to 8. As you see, as you go from lithium's got 3, 2 in the innermost and 1 in the second. Beryllium at 4 has 2 in the innermost, 2 in the second. Boron at 5 has 2 in the innermost, 3 on the outer, and so on and so forth as you go across the row until you get to neon, 2 in the innermost, 8 in the outer, and then we call that a full orbital if it's got 8 in the outermost ring, or if it's in the innermost ring, it's got 2. And that makes it very stable and unreactive. Anything that's got less than 2 in the innermost or 8 in the outermost is going to be reactive. Anything that has those full orbitals or full rings will be unreactive. And we call that last column with those unreactive elements, because they have full orbitals, the noble gases. Okay, here we're showing a couple of depictions that you may see in the textbook or on periodic tables or in other types of tools utilized in biochemistry, and this is generally what they mean. So here you've got He on the left side. In the middle, that's the symbol of the element. The number above, it's usually a whole number. That will be the atomic number, in other words, the number of protons. And then right below that, you have the atomic mass, which is the mass of the protons and the neutrons in Daltons. And on the right side here in that box, you've got a electron distribution diagram, which shows the nucleus in gray, which has your protons and neutrons in it, and then shows the electron cloud or the electron ring orbital on the outside with the 2 electrons in it to balance out the 2 positively charged protons in the nucleus. Again, you have your symbol up above it, You have 2 for the atomic number to the lower left corner, and then you have the full name of the element, in this case here, helium. So just depending on what table, different tables utilize different pieces of information. All the different periodic tables you can look at will contain some things in common, usually like the element symbol and the atomic number, but the other pieces of information they may contain can vary wildly depending on the size of the table and what the table is intended to be utilized for. So you generally make sure when you look at a periodic table, you utilize a periodic table that you make sure it's got the information on it that you need for your problems or whatever you're doing prior to actually using it. Okay, valence electrons are those in the outermost shell, or valence shell, that's the outermost ring. So, for instance, in that last table, if I go back here, let me see if I can go back here. Right here, that first shell, that's your valence shell, okay? Because there's only one ring, so that's the outermost ring. In hydrogen and helium, you've got 1 and 2, so those are valence electrons. When you get to the second row there, now the innermost ring is full, and there's a ring outside of that. That outside ring now becomes the valence level. So lithium has 1 electron at its valence level, the second shell, and beryllium has 2, and boron has 3, and carbon has 4, etc. And when you get to the third shell, now you have 2 inner shells that are filled. The first ring with 2, the second ring with 8, and then the third ring, the outermost shell, that now becomes the valence electron. So sodium has 1 valence electron, and magnesium has 2, and aluminum has 3, and so on and so forth. So what you see here is not only increasing numbers of protons as you go left to right across the table, and increasing numbers of electrons as you go left to right, but as you go top to bottom, you see that they have the same number of valence electrons. For instance, if you look down the first column, hydrogen, lithium, and sodium all have 1 electron in the outermost ring, and beryllium and magnesium have 2 in their outermost ring or valence shell. And boron has 3, etc. Carbon 4, nitrogen 5, oxygen 6, fluorine 7, and neon and argon 8. And helium only has 2 in its outermost ring, and that's the exception there for the first orbital. Okay, the chemical behavior of an atom is mostly determined by the valence electrons. Those valence electrons, those electrons in the outermost ring that have anything less than 8 are the reactive electrons. The inner full levels, the inner full rings with 2 or 8 generally do not react. Okay, so the outermost level, the valence electrons, are the ones that are going to be reactive in a chemical reaction. Elements with a full valence shell are chemically inert, like the noble gases, the far right column, because they have either 2 or in the outermost shell like helium, or neon and argon, they have 8 and everything subsequently below them. Okay, an orbital is a three-dimensional space where an electron is found 90% of the time. Each electron shell consists of a specific number of orbitals. So here, let's take a look at A. You have the electron distribution diagram. So it's showing neon here. It's got the gray nucleus, so it's going to have it's neon, so it's got 10 protons and neutrons there, all depicted by that round gray circle. That's your nucleus. That's got the 10 protons and 10 neutrons, let's say. And then you've got 10 electrons to match the 10 positively charged protons because it's a neutral element. So in the first ring you have, or the first orbital, you have 2 electrons, and then the second orbital you have 8 electrons for the total of 10 electrons. Okay, first shell, second shell. Now the first shell has a specific shape. Those 2 electrons in the innermost ring will actually have a spherical shape. They'll be in there circling around, flying around in the spherical shape. Okay, when you get to the second shell, now you've got 2 different types of orbitals. The first 2 electrons in the second shell, wherever they may be, make up this larger 2s orbital, and they're circling around in that. So you have 2 in the 1s, 2 in the 2s, and then the remaining 6, because remember 2 are in the so in that second shell, that upper right box, the second shell, there's 2 electrons in the 2s orbital, which means there are 6 in the rest of the second shell somewhere, and those are in what are called p orbitals. and there's orbitals along the x-axis, orbitals along the y-axis, and orbitals along the z-axis. Okay, and the reason this happens is because the electrons, remember, they're all negatively charged, and so if you think about magnets, things that tend to have the same charge repel each other, correct? And things that have opposite charge tend to attract each other. Well, electrons, since they are all negatively charged, are repelling each other, and they're all moving around rapidly, and they're all trying to stay as far apart from each other as possible. So what you end up getting are these kind of dumbbell-shaped orbitals, two along the x-axis, two along the y-axis, two along the z-axis, so it's three-dimensional. So when you superimpose all of these, neon's got 10 electrons. You'll get what's in diagram C there, the superimposed electron orbitals. That innermost, if you look at that picture, that innermost orbital there, that's the 1s with 2 electrons in it. And then you have the 2s, which is slightly larger, and that's got an additional 2 electrons in it. And then you have these 3 dumbbell shapes, one along the x, one along the y, one along the z-axis. Those are the 2p orbitals, and each of those lobes on the dumbbell contains an electron, so 2 along the x-axis, 2 along the y-axis, 2 along the z-axis, and there you've got 10 electrons. And that's roughly what the shape of neon would look like as far as its electron cloud, where the electrons are most likely to be found. Okay. Now, when we draw these in like space filling models and stuff, we usually just draw them as round circles in a roughly relative fashion as far as size goes. But again, that's just to make life a little bit easier when we're depicting these things in models. Okay, the formation and function of molecules depends on chemical bonding between atoms. Atoms with incomplete valence shells can share or transfer valence electrons with certain other atoms. These interactions usually result in atoms staying close together, held by attractions called chemical bonds. A covalent bond is the sharing of a pair of valence electrons by 2 atoms. In a covalent bond, the shared electrons count as a part of each atom's valence shell. Okay, so here we have hydrogen. You have 2 positively charged protons there in the nucleus depicted by the pink balls, and then you have the electron clouds there in the yellow. Okay, you can see as they start to move closer to each other, you start to get an oblong, or I guess it's an oblong type shape, or an egg shape there where they start kind of pinching in towards the center as they start to kind of form a bond, and then they form a bond between these two where the electrons are actually shared back and forth between the two. So this is hydrogen now. It's a molecule. You've got 2 of them formed together, covalently bonded. So it's H2, and you can see because the yellows kind of merged there, that's indicating that there's a structural bond between the two. Now, a molecule consists of 2 or more atoms together, held together by covalent bonds. Now, remember, covalent bond and molecule, always remember those 2 things have to go together. We wouldn't call something like sodium chloride, which is ionically bonded, a molecule. We might in the vernacular, but correctly, we wouldn't do that in science. When you say the word molecule, it means something specific. It means that it's bonded covalently. There are different types of bonds, and we'll cover a couple of them in this course. But for this point here, I just want to make sure that you understand when you use the term molecule, it indicates to somebody just by using that term that it is covalently bonded. Okay, so sugar is covalently bonded. We call it a molecule of sugar, but sodium chloride, which is ionically bonded salt, we wouldn't call a molecule of salt. We wouldn't say that. That would be improper in science terminology. Okay, and salt itself is actually a classification. We'll talk probably about that a little bit later on as well. So a single covalent bond or single bond is the sharing of 1 pair of valence electrons. And if you have 2 covalent bonds, we call that a double bond. It's the sharing of 2 pairs of valence electrons. There is such a thing as a triple bond as well. So if you see double bonds and triple bonds, know that they're covalently bonded, okay? They're not any other type of structure. They're not a hydrogen bond or a van der Waals force or an ionic bond or any of the other types of molecular interactions that exist. If it's a double bond or a triple bond, it's always going to be covalent. Okay, the notation used to represent atoms and bonding is called a structural formula. For here, we're going to show hydrogen bonded to hydrogen, so we put a little dash between the 2. This can be abbreviated further with a molecular formula, for instance, H2. Because we have the 2 hydrogens that are attached to each other, we can write it as a molecule of H2, and they've done so there in the last bullet. Okay, so here we're going to take a look at figure 2.10, name and molecular formula, electron distribution diagram, the Lewis dot structure, and the space filling model. So you probably have had chemistry at some point in the past, so you're probably somewhat familiar with this a little bit, so we'll kind of go through it rapidly. But here in the very first row, you have A, which is hydrogen, H2. And you got two hydrogens bonding, and they're showing the two electrons being shared covalently between the two of each other. You've got the Lewis dot structure in the third column to the top, and then you have the structural formula right below it, H to H with a dash in between, and then you have the space filling model, which is just showing you like if you're going to use a balloon or whatever to represent each element, it's going to kind of show you how they're merged together. Okay. In B, you have oxygen O2. Here it's showing you that you have a double bond between the two oxygens. There's two pairs of electrons shared between the two, and so in the the Lewis dot structure, you have two sets of dots between each of the oxygens, and you would represent that with a double bond or two dashes, or it looks like an equal sign between the two O's to indicate that there's a double bond between the two oxygens. And again, the space filling model would look very similar to hydrogen, but because oxygen is a larger atom, we depict the size just slightly bigger in the space filling model. Okay, water H2O. Now you have hydrogens bonded to oxygens, and oxygens sharing electrons with hydrogens, etc. So you've got your electron distribution diagram there showing you've got the 8 outermost electrons in the oxygen, making it stable, and 2 for hydrogen, because hydrogen only gets 2 in its outermost shell. So it shows the H2O structure there. You have the Lewis dot structure showing the sharing of electrons between the hydrogens, each of the hydrogens and the oxygen. You have the dashes represented below because there's only one set of bonds between the hydrogens and the oxygen, so it's just a single bond or a single dash between each hydrogen and the oxygen. And then you have your space filling model kind of showing the relative size of each of the atoms compared to each other and roughly how it's kind of bent in three-dimensional space. Finally, in the fourth row, you have D, methane, CH4, where you have 4 hydrogens bonded to a carbon. You have the Lewis dot structure that shows the pair of electrons shared between the carbons and hydrogens in each of the 4 cases, and then represented in the structural formula with a dash. So you have carbon to hydrogen 4 times, each one represented by 1 dash, and then you have the space filling model with the black carbon in the center and the 4 hydrogens in light gray around it. So you can see how they're depicted, and again, they're trying to, you notice they're not all one on each of the 4 sides. There's like 1 on the top, and then like the 3 on the bottom set up almost like a tripod. Remember, again, they're all negatively charged electrons in these electron clouds, so they're trying to stay away from each other as far as possible. So it doesn't have like a plus sign configuration where you have a hydrogen on the top and bottom and 1 on the left and right. You actually have like 3 on the bottom like a tripod and then 1 up on top, so they're just as geometrically far apart from each other as possible. Okay, bonding capacity is called the atom's valence. Covalent bonds can form between atoms of the same element or atoms of different elements, and a compound is a combination of 2 or more different elements. Atoms in a molecule attract electrons to varying degrees. Electronegativity is an atom's attraction for the electrons in a covalent bond. The more electronegative an atom is, the more strongly it pulls shared electrons towards itself. In a nonpolar covalent bond, the atoms share the electrons equally. In other words, they don't spend more time on one than the other. They are equally shared. It's nonpolar. In a polar covalent bond, one atom is more electronegative and the atoms do not share the electrons equally. Unequal sharing of electrons causes a partial positive or negative charge for each atom or molecule. So let me talk a little bit about polar and nonpolar. Nonpolar means it's kind of like a battery where you have a plus end and a minus end. Okay, you have a slightly different charge on each end. They're not similar. Nonpolar means you kind of have the same charge all the way around, whether it's all positive or it's all negative. It's like a battery that's got pluses on both ends instead of a plus and a minus. Okay, in which case I guess it wouldn't be a battery, right? It'd just be something with 2 positively charged cations or 2 negatively charged anodes or something like that. But bottom line here is polarity is going to be like a north pole and a south pole. You have a plus and a minus side, and nonpolar means you have the same, whichever it is, positive or negative, it's going to be the same all the way around it. Okay, so here they're showing a water molecule. What they're showing is oxygen's negatively charged, and it's got kind of strongly negatively charged. So it's got a partially that squiggly looking Greek letter there. That's a indicates that it's a partial charge. Okay, that's the symbol for a partial charge. Kind of almost looks like a snake ready to strike. So oxygen's got a partially negative charge, and hydrogens are positively charged. They got a partial positive charge. So you can see on the top of the water molecule you have a negative charge, and on the bottom you have a kind of positive side. So that creates polarity. Ionic bonds. Atoms sometimes strip electrons from their bonding partners. An example is a transfer of an electron from sodium to chlorine. After the transfer of an electron, both atoms have charges, and these are called ions. So this is called an ionic bond. So here you have sodium and chlorine. Like we said before, sodium's a soft, buttery metal that you can cut with a butter knife, but it's also rapidly oxidized in the presence of air or oxygen, and it'll catch on fire and explode in short order. Chlorine's that yellowish green gas that if you inhale it, it's going to strip hydrogens away from water molecules in your lungs and form hydrochloric acid, and it'll burn your lungs out. So both very dangerous, but here's what happens. In sodium, in the outermost of valence level, you have 1 electron, and in chlorine you have 7. Both of them kind of want to get to 8, right? They want to look like they have 8 on their outermost ring. So if sodium has its 1 electron in the outermost ring stripped away, then the next innermost ring is going to have 8, which makes it stable. And chlorine likewise has 7 in its outermost ring. It's looking to pick up 1. If it gets that 1 from sodium, it will then have 8, and it will be stable as well. And so this is what you have. Sodium loses that outermost electron, so therefore that ring disappears, and now its outermost ring has 8 electrons. And chlorine picks up that 1 sodium electron from the outermost ring, and it now has 8 electrons in its outermost ring. You have a bond between the two. This is called an ionic bond because one is now positively charged and the other is now negatively charged. And so we call that an ionic bond. The positively charged piece is called the cation, and the negatively charged piece is called the anion, or as I like to remember it, a negative ion, anion. Okay, a cation is positively charged, anion negatively charged. An ionic bond is an attraction between an anion and a cation. Compounds formed by ionic bonds are called ionic compounds or salts. Salts such as sodium chloride, table salt, are often found in nature as crystals, such as this, in which case the sodiums and the chlorines alternate with each other, giving it its crystalline structure, that geometric shape that you see in a crystalline crystalline salt crystal. Now, when we talk about weak chemical reactions, we'll find that most of the strongest bonds in organisms are covalent, and they're formed in a cell's molecules, but many large biological molecules are held in their functional form by weaker bonds. The reversibility of weak bonds can be an advantage for living organisms. A hydrogen bond forms when a hydrogen atom covalently bonds to one electronegative atom, but it's also attracted to another electronegative atom. So it's got to be bound to one already and weakly or attracted somewhat to another electronegative atom. And that makes it a hydrogen bond. And this is for hydrogen. In living cells, the electronegative partners are generally oxygen or nitrogen atoms, where you'll have an oxygen already bonded to a hydrogen, like in a water molecule, that's also bonded to another hydrogen. Okay, so here you've got a water molecule on the top with its partially positive and partially negative sides. You have an ammonia molecule at the bottom, which is nitrogen with 3 hydrogens with its partially positive side, partially negative side. Now notice the partially negative side of the nitrogen itself, the blue, is attracted to the partially positive hydrogen, which is also already attracted to another electronegative atom, the oxygen. Remember, the hydrogen bond has to be a hydrogen atom attracted to a existing already positively charged hydrogen attracted to a already electronegative atom. So you have nitrogen attracted to the partially charged hydrogen, which is already attracted to the partially charged negative oxygen, and that forms what's called that hydrogen bond. Now, van der Waals interactions, if electrons are not evenly distributed, they may accumulate by chance in one part of a molecule. Remember, we said electrons are always trying to stay away from each other as far apart from each other as possible, but periodically there are intermittent, very brief moments where they tend to be more of them on one location than another as they're moving around rapidly, and that can cause some temporary polarity. So van der Waals interactions are attractions between molecules that are close together as a result of these charges. Now, collectively, such interactions can be strong, as between molecules of a gecko's toe hairs and a wall surface is why geckos can walk up walls. Molecular shape and function. A molecule's size and shape are key to its function. A molecule's shape is determined by the positions of its atoms' orbitals, and in a covalent bond, the s and p orbitals may hybridize, creating specific molecular shapes. So here they're showing the 3p orbitals can kind of hybridize into modified orbitals, which would create 4 hybrid orbitals, the 3p orbitals creating 4 hybrid orbitals, and giving it a shape like a tetrahedron. Molecular shape determines how biological molecules recognize and respond to one another. Opiates, such as morphine and naturally produced endorphins, have similar effects because their shapes are similar and they bind to the same receptors in the brain. So here, if you look in the box at the natural endorphin, they're looking at that region that's in the little gray or the little black text box there, and they're comparing it to morphine, right? So morphine's a man-made chemical that's a drug. It's got a receptor that's similar in shape and function that can actually bind to the endorphin receptors in the body as depicted in the lower picture. And so the natural endorphin can give a particular stimulus or a particular response at the endorphin receptor because it can bind there. And morphine can do the same thing because morphine's got the same shape, man-made, but same shape. It can bind to that same receptor and simulate. Okay, chemical reactions make and break chemical bonds. Chemical reactions are the making and breaking of chemical bonds. The starting molecules of a chemical reaction are called reactants, and the final products of a chemical reaction are called products. So here on the left side you have hydrogen atoms reacting with oxygen to produce water molecules. Okay. Photosynthesis is an important chemical reaction. Sunlight powers the conversion of carbon dioxide and water to glucose and oxygen. If you look here in this chemical reaction formula, you have 6 carbon dioxide plus 6 water molecules produce 6 glucose molecule, or excuse me, 1 glucose molecule. There's no value in front of the C6H12O6, so it's just 1 glucose molecule and 6 oxygens. And that's the chemical formula for photosynthesis. Okay, here you can see, remember that reaction here, you got glucose and water, or excuse me, glucose and oxygen produced in the chemical reaction to the right side of the arrow. And here's the LOD leaf where you can see the oxygen being produced as a result of that photosynthetic reaction in water. Now, all chemical reactions are reversible. Products of the forward reaction become reactants of the reverse reaction. The 2 opposite-headed arrows indicate that a reaction is reversible. You will see these from time to time. Chemical equilibrium is reached when the forward and the reverse reactions occur at the same speed or same rate. At equilibrium, the relative concentrations of reactants and products do not change. And that's it for Chapter 2. Thank you for listening, and we will talk very shortly. Thank you. Bye-bye.